Today we have Dr. Betsy Wells from DWRs talking about getting to the bottom of it, which she's going to talk about the Benthic monitoring program. So Dr. Wells is currently an environmental scientist with the DWR. She's working on the Benthic monitoring program. Please visit this website listed here for more information. She has a bachelor's degree from Williams College, moved over to the other side of the country, and did her PhD at the UC Davis. Worked on the marine invasive species and the species interaction and prey-predator interaction in the evolutionary history. I don't know what that exactly means, but it sounds pretty important. So this is what I know of the benthic macroinvertebrates. Obviously, you guys haven't seen this because this is from eastern mountain streams, which is a good indicator of higher water quality against lower water quality. Although I know Dr. Wells is going to talk about a little different stuff. So for that, I'm going to give the podium to her, and she's going to tell you what she's been working on these days.
Hi, thanks Wasong for the introduction. Thank you all for coming today. I'm really excited to see so many people who are interested in benthic ecology, even if just as a curiosity. And thank you also to the Delta Science Program and SWAMP and the other group, I'm sorry, was it the Environmental Restoration Program, who hosted the Brown Bag Seminar. This is a terrific series. I'm so glad to be a part of it. Thank you all for hosting me today. Wasong said I'm going to be talking about the EMP's benthic monitoring program in the Delta. I know a lot of you here are pretty familiar with some of the Delta science, but I realize some of this may not be familiar, so forgive me if I'm shooting a little bit lower on some of you and do some of the introductory biology. I'm sure there are some people listening who would appreciate defining some of the terms as we go. So as a roadmap for my talk today, I'm just going to go through, number one, what is benthic ecology? Why we care about it? Why would we study it at all? I'm going to talk about some of the role of bioindicators and the role of the benthos in the food web and some of the policy behind why we studied the benthic ecology of the Delta. I'm going to go through some of our sampling protocols and some of the data that we get out of the sampling. And then I'm going to try to give some form to this massive data by talking about some of the trends, whether the seasonal trends or the geographic trends, to help you guys come away with a picture of if you're out in the Delta looking down through the water and wondering what is under me, you can have a fair idea by the end of this talk of what you might be finding. And then I'll talk about some of the other bigger and deeper analyses that people have done with the benthic data that we've gathered. And just before I go any further, can the folks in the back hear me okay? Are we doing okay? Terrific, thanks. So to start with, what is benthic ecology? When I tell people I'm a benthic ecologist, their eyes kind of glaze over. And then the next thing they ask is, what's benthic ecology? So essentially, it's whatever lives at the bottom of a water body. However deep, whatever sediment, it's what lives at the bottom, at the interface of the water and the sediment, whether in the sediment, on the sediment, associated with the sediment. And I just want to point out, through sort of a happy coincidence, tomorrow is the start of the National Benthic Ecology Meeting in Portland, Maine. And I don't just want to plug this conference, although it's a great one, but their conference flyer actually had a terrific illustration of some of the variety of stuff you would see in the benthos, everything from seagrasses and seaweeds to oysters and mussels and sea stars and cod and lobsters and scallops. I mean, you get this massive diversity and variety of life forms in the benthos, more than a lot of other ecosystems, which is one of the things I find so exciting about it. And I just wanted to point out, here's a great picture of some of the stuff we could be looking at if we were in Maine. So like I said, the benthos is at the bottom of a water body. So 70% of the Earth, roughly, is covered in water. That means that 70% of the Earth is covered in benthic habitat. So before you think of the benthos as sort of just a niche thing to study, by surface area, benthic ecology is one of the bigger things out there. And it's everything from very shallow. This is a picture of a Chinook salmon in a red up in Washington State, all the way to very deep. And the picture of that submersible is James Cameron's submersible that went down to the bottom of the Mariana Trench. All of those are benthic habitats. They have nothing else in common, but they are benthic. So, okay, that's what benthic ecology is. Why does anyone care? After all, it's down there, right? And the first answer, sort of classic answer, is that benthic communities are a really classic bioindicator. They're a proxy for how the health of the water body is doing. A lot of people who've worked in streams, this is primarily how they know them. They look at the benthic community as a sense of how the ecosystem and the community as a whole are doing. And there's a couple reasons for this. Number one, like I said, they're the animals that are associated with and in the sediment, so they're geographically stable. If you study fish, you know pretty well fish move. If you study zooplankton or phytoplankton, they move as well, either with the currents or passively drifting or stuff like that. If you're buried in the sediment, you're more geographically stable. That means if I go out to a spot with GPS, I can go out again the next month and I can sample that same community and know that that has a relationship to the community from a month before, which is really convenient as a researcher. The other thing is that they actually are on a really convenient time frame. They're not too short and they're not too long. So if you've got a response to some sort of input or disturbance, whether it's a chemical change, it's a pollution plume, it's a change in salinity, it's a deoxygenation event, or a biological change like a new invader, a predator that's come through, you usually get a time response. You usually get a response in the benthic community of a few months to several years. Of course, there's outliers, but a few months to several years both lets you go out and document changes that happen as well as the response from changes that happen. Things that respond really, really quickly, like phytoplankton, blink and you miss them. Things that respond really slowly, like redwoods, you could be waiting for decades to see the response and the recovery back from, say, a forest fire. So a few months to a few years is really convenient for human and funding timeframes. So for that reason, they're also a great thing to look at as a community indicator. And the final thing is that benthic communities tend to be very highly diverse, not just in number of species, but in ways of life. So you get everything from, as you can see, this is a picture of an Antarctic benthic community. You get worms, you get clams, you get snails, you get fish associated with the bottom, you get crabs. You get all of these, pretty much any way of life, whether predator, scavenger, filter feeder, living in the sediment, living on the sediment, being parasitic. You find it all in the benthos. I had a terrific undergrad professor, Jim Carlton, who basically said that anything you can find in sci-fi, you're going to find in benthic invertebrates. And so this means not just that it's cool to look at, which it is, but it does mean that you get a very nuanced, very sort of fine-grained look at how communities are responding, because all of these different ways of life and all of these different species will have different responses to the different inputs. So you can actually get, as opposed to, say, a guild of fish that all do the same thing, you can get a very finely nuanced set of data from the benthos, which is really useful to us. Okay, great, you can use them as bioindicators. Well, how? Those of you who have studied streams like Wasong are sort of going, yes, we know this. But basically, you measure some things about the community, whether it's species richness, the biomass, which is the weight of the living organisms there. You look at the functional diversity, if you have all predators or all grazers or a bunch of both. You look at if you have a whole bunch of pollution-tolerant species, which may hint you've had some pollution in the past. You look at whether you've got pollution-sensitive species, which sort of indicate, hey, this place hasn't been all that polluted. You measure a whole bunch of different metrics and come up with sort of a combined index of all of these things and come up with sort of a final grade for the place you're looking at. This is a map of the Chesapeake Bay, a place very dear to my heart. I worked on it for about 5 years, where they've done a benthic index like this. And it's a classic heat map. The green areas are rated sort of a healthier benthic community, and the red ones are less healthy. And it's a really good visual way of saying what is the overall health, or at least a proxy for the overall health of a water body. There is an alphabet soup of different indices. Some of them are really designed for streams, some for rapid assessment, some are adaptable to marine or lakes or whatever. And you can sort of pick and choose and decide which ones work the best for you. And people have done this in San Francisco Bay and the Greater Bay. A couple of papers within the last 10 years here, where, for example, they're comparing, how different experts agree on the benthic index, or they're comparing the different benthic indices and see how well they agree. And you can come up with some pretty decent estimations of, okay, this area is impacted, this area is more pristine. Unfortunately, now that I've set up benthic indicators is so useful, I'm going to knock them down. They don't work that well in the upper estuary, and I wish I had a definitive answer why. The best answer I can come up with is that, number one, the delta as a whole tends to be largely changed. I mean, it's a very altered ecosystem. There aren't a whole lot of pristine places left to serve as reference sites. The other thing is that it's a very alluvial system where you don't have a lot of points of disturbance, like an outfall, like you do in the bay, or you do have dredging, but the impacts tend to be really widespread. So when you try to say, okay, this place is impacted, what would it look like if it were pristine? It's very hard to say why, and it's very hard to come up with a good reference site. And the other thing is that, as I'm going to talk about a little bit later in the talk, the delta and the estuary are so variable, mostly because of the salinity gradient. That trying to compare Sassoon Bay to Clifton Court 4 Bay, where the pumps are, I mean, they don't have anything in common. So it's not even comparing apples and oranges. It's like apples and arthropods. It's really hard to come up with reference sites. So anyway, it would be really cool. We can probably come up with some other indices, but for right now, benthic indicators are not high on our list of what to do. Okay, so we don't care about them because of benthic indicators. Why do we care? Food webs. Food webs are important. And in almost every estuary food web you look at, the benthos is a central and critical part of the larger food web. This is a diagram actually from New York's Plum Island ecosystem. And what I like is that it actually is a really nice, clear diagram of a tidal river. A lot of the a lot of the species here are representative of species that we get here in our estuary. And what I especially like, because I have a lot of background in invasive species, some of the species are actually the same ones too, because they came here. So for example, Rithropanopius, this little crab, black-fingered mud crab, we got it. Moroni sectalis, striped bass, we got it. So I just wanted to point out the similarities you see in this diagram, we're going to find them in the delta too. When you look at some of the conceptual models of delta food webs, again, benthics are central and are often a major pass-through point from primary productivity through secondary productivity and up to the things that a lot of other people care about, like fish and birds. You either have to go through the planktonic route or you go through the benthos. They contribute a lot to the food web. If you've ever gone fly fishing, you can tell that chironomids, these are little midge and fly larvae up here. There's a lot of tide flies that look a lot like those, and there's a reason for that. A lot of fish like them up in the fresh water. Oligochaetes, a lot of fish and other invertebrates eat the worms that you find at the bottom. Amphipods, essentially if you think of them as mini shrimp, you're doing okay. These little arthropods, also a major part of a bunch of different fish diets. And clams, I mean, birds, fish, other invertebrates like crabs, everyone seems to like clams. So they contribute a lot to the food web. And there's been a fair chunk of research devoted to the place of the benthos in the food web, whether it interacts with phytoplankton, with bacteria, how they interact with fish diets, how the fish diets have changed in relationship to benthos. And possibly my favorite paper title of all time, for those of you who can't read it, it's Overbite Clams, Corbula amurensis, Defecated Alive by White Sturgeon, Asapenser transmontanus. Failed predation sometimes just as awesome as successful predation. And I'm going to tell a very simple story to illustrate some of the place of the benthos in the food web about a very complex phenomenon. Most of you have probably heard about the pelagic organism decline. This was a phenomenon starting in about 2002 where multiple fish species at once started to go into nosedives in terms of the populations. And people had a lot of interest in why this was happening. These four fish especially were the focus of a lot of the pod research. And they have different ways of life, different life histories, what could be causing this. And like I said, this is a very complex phenomenon. The benthic food web portion is only a small part, not necessarily even the biggest part, but it is a part of it. And so I just want to illustrate that part of the pod. There's lots of other things like habitat, chemical influx, top-down pressures like salvage and predation that also contributed to the pod, but the benthic part's of interest to me, so that's what I'm going to talk about. So here's a really simplified delta food web. Fish on top, eating invertebrates, like these are mysid shrimp, who in turn eat things like phytoplankton and zooplankton. You get more phyto and zooplankton, you get more mysid shrimp, you get more fish. The delta is happy. Oh wait, 1986, Potamacorbula amurensis. This is the Asian overbite clam. It's so called this because one shell is a little bit bigger than the other, so it actually does have a little overbite. Invaded San Pablo and Sassoon Bays in 1986, and this is an extremely efficient filter feeder. It really hoovers out a lot of the phytoplankton from the water column, and it grows in incredible densities. We regularly get tens of thousands per square meter on the bottom. I think our record so far is north of 150,000 per square meter. Granted, most of those were really little, but the densities are pretty amazing, and they just are, they eat a lot of the phytoplankton and take the food web kind of out at the knees. When you look at some of the research that has followed the invasion of Potamacorbula, you really see this. I've marked with red arrows 1986 where the clam came in, and you can see this is chlorophyll A levels in Sassoon Bay and then the delta at large. And you look, after 1986, a lot of them go down pretty dramatically, especially in Sassoon Bay, to where they are today, which is pretty low levels. You look at the mysid levels, and this has been tied pretty firmly to the loss of phytoplankton brought on by the clam. The mysids plunged. And so again, this is only part of what may have contributed to the decline of the fish. It's not exactly a smoking gun, but it is probably part of the overall picture. So the benthos is a big part of the food web, and it can have these effects that ripple up through the food web. And finally, another reason for monitoring the benthos is there's policy that tells us to. Water Right Decision 1641 mandated comprehensive environmental monitoring. This is performed by the Environmental Monitoring Program, which is under the IEP umbrella. And benthic monitoring is part of that. So we perform this in tandem with zooplankton, phytoplankton, and water quality monitoring. It's part of this integrated program. Okay, well, how do we do this? Well, what happens is we go out monthly to each of 10 different stations from San Pablo Bay in the west all the way into Stockton and Clifton Court 4 Bay. Currently, we're monitoring at 10 different stations. We've been to up to, in the past, going back to 1975, which is when this whole program started, we have a total of 53 stations that we've gone to. Only 10 of them are currently active. But we've got a lot of past data, historical data, that's snapshots in the past. And the current 10 stations we've been monitoring for a solid 20 years. So 20 years by itself is a pretty nice, solid data set, and you can extend it back further with these historical stations. What this looks like is we get on a boat, go out in the field, and we have a ponar. I'm not sure how many of you have seen this, but it's basically a big double-barreled dredge with 2 scoops on a rope. We lower it down, it hits the bottom, it releases, takes a double scoop of sediment, we pull it back up, sieve it out in the field, pour the animals into a jar and preserve them, and take them back to give to our taxonomist. In the field, our taxonomist goes through, sorts them, counts, and identifies every single organism down to a ½ millimeter large. This is a big job. And what they look like in the end is a lot of small vials filled with very small organisms, or in some cases, like these guys, much bigger clams. And the reason they're pink is because we add rose bengal to the preservative solution, because if you're trying to pick out small, white, translucent, or brown-colored things against a background of sand and mud, it really helps if you dyed them shocking pink instead. It really helps pull them out. So all of our samples are bright pink. And I just want to mention right now, for the entire 40-year history of this project, we've had a single taxonomist, Wayne Fields. I don't know very many data sets that have had this continuity of information input and this high level of information input. It's a pretty amazing data set, and Wayne has been a massive part of this effort through the 40 years. Okay, great. We've got our samples. What do we get? Everything. Most of the invertebrate tree of life, we get at least a few samples from every branch. Right now, regularly, we get specimens from 11 different phyla. That's sort of an amazing variety of functional group, of body form, and of way of life. And I realize not everyone loves invertebrates as much as I do, so I'm just going to give quick descriptions of what these are. Annelids are your segmented worms. Arthropods are everything with a hard exoskeleton and joints, so crabs, shrimp, amphipods. Chordata, it's vertebrates, but it's also sea squirts, which are our closest relatives. Cnidaria, we get a lot of anemones. We do get jellyfish, but we don't identify them because they don't tend to end up in good shape after all that they've been through. Echinoderms, we do get brittle stars and sea stars. Mollusks, that's everything from the snails and the clams and the mollusks and the mussels. Nematodes are the unsegmented roundworms. Nematomorphs are like nematodes, but they tend to be parasitic. Nemerteans are ribbon worms. Phoronids are called horsehair worms, but they're actually their own weird phylum. And platyhelminthes are flatworms. So we're kind of worm heavy here, but like I said, it's a big variety in terms of all different ways of life. However, when you look at the distribution, it's dominated by the arthropods, by the annelids, again, the segmented worms, and the mollusks. And then you get this little slice, and this is from about a decade of data, you get this little slice that's everything else. So like many biological distributions, you get a few things that dominate numerically, and then a bunch of rare things. And I just wanted to show you some of the pictures, because I know this is sort of hard to imagine, but one of the things I wanted to point out, so it's just, again, you get a bunch of different ways of life. So in the center, we've got Tamacorbula, that's our overbite clam. We get snails, we get these oligochaetes, which look like earthworms, but live at the bottom of the water. We get mussels and amphipods. This is Granditorella, it's one of our more common amphipods. Again, think of them as small shrimp. Another one I want to mention is Corbicula, which is another invasive clam. What's kind of cool about Tamacorbula is that it inhabits marine to brackish waters, and then Corbicula does freshwater down to brackish, and the two of them sort of cover the waterfront in invasive clams. We get snails and humations and other kinds of clams. So this is a pretty fair sampling. I took these pictures last month, so if you were to go out in the Delta right now, this is the kind of stuff you're going to see right now. And the main product that we're sampling is the data, the where, the when, the what, how many. Just the basic data of what we found when we were out there. And all of our data for the last 40 years is available right now, publicly. You can go back to your desks today and download it and play with it. We have up through 2014 up. We're finishing up doing QA/QC for 2015. That should be up soon. And all this data is up there. Go have fun with it. Tell me what you find. I'd love to hear. People access this all the time and want to know, like, hey, have we found any other stuff? And yeah, it's terrific to have all this data that's publicly accessible from all of these years and all of these sites. And hopefully in the next year or so, we're going to have a new web portal where not only do we give you some more sort of context for the benthic invertebrates, we introduce you to some of them, we tell you some of the data stories, but hopefully we'll be able to have the data interactive. So you don't have to just download it all and then load it into Excel or whatever. You can actually go select your species and your dates and your times and play with them and sort of get some idea of the patterns on your own in an interactive way. So that's our hope to get that up in the next year or so. Keep your eyes peeled. This will also be happening for phytoplankton and zooplankton and stuff like that. So pretty exciting. Okay, got 40 years of data. Let's try to condense it down. We found a total of 460 species. I just want to point out there's only about 55 fish species in Delta, so yeah. We've got 140 separate individual taxonomic samples. So think of each of those as a vial. We've gotten 140,000 of those, and nearly 6 million individual organisms over 40 years. It's a lot of data. And okay, so I'm going to try to give you some patterns to think about to try to organize that, because that's too much data to hold in your mind at any one time. And the first thing is there's really, really strong seasonal patterns. As you might expect with sort of short-lived things, you're going to find some strong seasonal patterns. This is one site. This is D6. This is right by the Benicia Bridge, just upstream of there a little bit. If you know where the mothball fleet is, it's right off that kind of deep site, very muddy. And when you go out there, this is just from 2014. It was a nice representative year. What you see is that you get a massive peak in Tamacorbula recruitment that peaks in late summer, early fall. And in some years, it's an order of magnitude larger in the peak recruitment times than in the rest of the time. I mean, that's just a huge change. And the reason is they reproduce in the summer, and you get all these little ones that eventually get weeded out or eaten or whatever, and then it falls again in the winter. So that's the big numeric pattern. If you go out this time of year, you're not going to find very many. Go out in May, we're going to be picking a lot more of them out. In August, we'll pick a lot of them out. And the other part of this is, I just want to point out, this is actually the same graph. The right-hand graph is just everything that was under 500 organisms per meter squared. So it's this tiny little line at the bottom, just blown up on the right-hand side. I just want to point out also, Tamacorbula is really numerically dominant to everything else. But I just want to point out, these are some of the other dominant species out there. You get a couple of different amphipods, Ampolisca and Coryphium. You get a sea squirt, and you get a barnacle, Balanus improvisus. And all of those have their own distinct patterns. So you can go out 1 month and go out 6 months later and see almost nothing the same. in those 2 different months. That's one of the reasons that we sample every different month, because some of these have really sharp peaks, like Ampelisca. If you look at the right-hand graph, that red peak, you know, it really only peaks in November. If we didn't go out in November, we wouldn't see it that year. So there's a really strong seasonal pattern at a lot of these sites. Some of them are there year-round, but not. But the other thing that I really want to emphasize is the main driver at all of these sites is salinity. Salinity is the master variable. This is not a surprise to anyone who's listening today. We all probably care a lot about salinity. This is a nice model of the low salinity zone by Michael McWilliams that was done for DWR. And it's just to show that, yeah, salinity, in fact, does get lower as you go upstream on a roughly west to east gradient, which is convenient for us. And so here's some of the data. This is sort of the last decade of data sort of aggregated together. And I just want to show you that I've on the bottom axis here of this graph, I've organized the sites from highest salinity on the left-hand side to lowest salinity on the right-hand side, which again is roughly west to east. As I said, really convenient to look at. And if you look at the sites in San Pablo Bay, you get a very distinct marine, or at least, you know, polyhaline clade that as you get up into Sassoon and Grizzly Bays, a lot of those just drop out. You just don't see them there. Instead, you get Potamacorbula taking over and being the numerically dominant bivalve up there. And as you move further up to the confluence and up into the Sacramento and San Joaquin rivers, all of a sudden what you're getting really is a couple of really truly freshwater clams, Corbicula and Pisidium. So you get this complete changeover in species identity because of salinity. There's 0% overlap at the upper and lower sites. When you look at some of the arthropods, it's the same pattern. You get these marine clades down at D41, D41A in San Pablo Bay. It shifts up into the stuff you find more in Sassoon Bay, and then you really shift into a freshwater assemblage as you go upstream. It's rare that you find a single variable explaining so much of the variability, and I just want to point out this is a massively, overwhelmingly important driver for the benthic community here. And with wet and dry years, as you can imagine, that changes. You get that sort of weeks, sort of months to years response to an input. The other thing that I'm not sure people have looked at a huge amount in the Delta, but I sort of noticed it and wanted to point it out to you guys. I have a lot of interest in native and non-native species because I've done a lot of work on non-natives and invasive species. And one interesting pattern I saw was when you look at 3 of our longest sampled sites that go back to 1980, you find more as you go downstream. So you find a lot more in Sassoon Bay. This is just the number of non-native species that we found in each year. And you get fewer as you go upstream. And I'm not totally sure why. I'd love to talk to people who are interested about this. I don't know if it's just the lack of vectors. You get a lot more shipping in San Francisco Bay and Richmond. So you get more ballast water coming in and maybe introducing new vectors. You do get shipping, of course, up into Sacramento and Stockton, but not the same number. Maybe it's just that the upstream is not as hospitable a site to new arrivals. Not sure. It's kind of an interesting puzzle. When you look at the number of non-native individuals, sort of you count every head in the population, it's even more pronounced. Of course, D7 is one of the spots where you see a lot of Potamacorbula, the invasive clam. So, of course, most of it is dominated. From 1986 onward, it's really clear shift to being dominated by non-natives. And you don't see it as much upstream. Anyway, kind of a cool pattern. Kind of wanted to throw that out there for other people to think on, too. Another really strong pattern that we've seen very, very much the last few years of the drought is that water year has a big effect, but more on downstream than upstream, which is kind of a reverse way of thinking about the Delta because people think about, wanting to keep the freshwater areas fresh. But if you're benthic, as long as it's fresh enough, you don't change that much. But downstream, it really matters. So I don't know how many of you have worked with MDS plots before. Essentially, I'm not going to go into the mechanics of them, but all you have to do is know that each point here represents a month of data for this one site in San Pablo Bay over the course of a decade. The sites that are closer together are more similar in the number and identity of species that we found. The sites that are further apart are more divergent. You find different species, you find different numbers. And I just want to point out there's a really strong wet year to dry year pattern. The spots over in sort of the lower left-hand corner are 2014 and 2013, the really dry years. When you compare them to 2016 and 2011, the wet years, they're much further apart. In fact, I looked at sort of the percentage dissimilarity, and you get 98 to 99% dissimilar. It's as though it's an entirely other site between wet and dry years. It completely flips over. I'm actually really excited to see what happens this spring after all this rain we got. We've been seeing a lot of fully marine animals in San Pablo Bay this year. I think they're all going to go away, and we're going to get a whole bunch of more brackish water species showing up. It's going to be, I mean, yay for water for a lot of different reasons, but just from a data point of view, I'm really excited to see what happens. So, okay, water years are very important downstream. How about upstream? Not so much. If you look at C9, Clifton Court Forebay, you do see quite a spread in terms of, you know, some years and other years, but it's not according to water year. And I don't know exactly why, if it's pumping patterns or temperature or nutrients. I'm not totally sure what explains this pattern. There is quite a spread, but it's not water year. So that's actually kind of a cool thing. Again, like to talk to more people about this. But I just want to emphasize, when you look not just at the two extreme stations, but when you look at the whole range of our 10 stations that we're currently looking at, and you ask how similar are they to each other, they fall out into groups really according to salinity. San Pablo Bay sites really group together. Sassoon and Grizzly Bay are their own clade. You know, Confluence really is its own thing, and then you get the rivers that are all kind of like each other. And again, it's by salinity. It really is the major driver. If you're like, wow, these trends are really cool, I can't take notes fast enough. I want to read about them more at my leisure. We actually publish status and trends every year on the Benthic monitoring in the IEP newsletter, and actually the current issue right now has the Benthic monitoring write-up from 2014. Where I not only talk about some of the salinity and the seasonal trends, but I talk about how they compare to some of the historical trends. How is, you know, San Pablo Bay this year different from San Pablo Bay over the last decade or so. So if you're interested in this, there's a nice little write-up about it in the IEP newsletter. I encourage you to go look at it. I'm not the only person to notice this, obviously. There have been some really interesting papers, people looking at groupings along the salinity gradient. This is Heather Peterson and Mark Betsy in a 2010 paper where they took not just the last decade of data, but they took 27 years of data. And they asked not just how do the sites group together, but is salinity explicitly the driver? And the answer is yes. Yes, you do get these same groupings, sort of San Pablo Bay, Grizzly Bay, the Sacramento River, Old River. They all sort of really group together according to salinity and geographic location. And 97% of that variability is explained by mean annual salinity, which is kind of a blunt tool, but it explains a lot of variability. So what if you expand your range even bigger? This is Bruce Thompson's work from 2013, where he took not only DWR monitoring data, but I think 7 other data sets of benthic data, and basically said, does this pattern hold for the rest of the greater San Francisco Bay and estuary? And yeah, it does. He actually found some very cool groupings based, again, if you look at his key, oligohaline, mesohaline, polyhaline. So it really is salinity that's organizing the benthic community throughout the greater San Francisco estuary, which I think is just a really neat extension of the monitoring we're doing. I'm so glad that, you know, that our data contributes to papers like this. Very cool new extension of our work. This is a collaboration with USGS that just came out a month or so ago. So the folks at USGS are kind of curious about modeling water and hydrodynamics and phytoplankton flows, and they were asking, can we have the counts of all of the clams that you've ever collected in the past, especially those 2 invasive clams, Potam corbula and corbicula. Can we estimate how much they were grazing in the past? So they actually went through our historical samples, pulled out every clam, and measured it. A fair number of those, almost 6 million organisms, were those clams. So that's a lot of measuring. And they converted those measurements into biomass and then grazing rates. And they've actually come up with estimates that could be used for big modeling projects. So if you're trying to model like phytoplankton supply in the delta, and you want to know what's the minus term of stuff that got eaten and lost to the benthos, this work is going to help supply that. So that's actually really neat that some of our historical data got used for that. And Jeff Crowder is actually going to be speaking at this at the IEP workshop in April. highly encourage you to go listen to that. Should be a good talk. I'm going to steal just a little bit of his thunder right now and present one of the graphs from this paper. This is from 1987, and I just want to point out this is the year after the Tamacrobial invasion, and there's already quite a big amount of grazing pressure in the Carquinez Straits and in the lower Sassoon Bay, much bigger than Corbicula flumineo, which had been there for many years. So this is the kind of stuff Jeff's going to be talking about. I just like seeing this, our count data being expanded into this sort of more effect of the benthos on the food web. Another sort of a really interesting effort that we did last year, this was the State of the Estuary report, which was spearheaded by the San Francisco Estuary Partnership. They were basically asking, what is the health of the bay? And they wanted to have a bunch of different indicators, everything from waterfowl, herons and egrets, flow, is the water safe to drink? And in wildlife, they basically went to a lot of data sets and a lot of experts in these groups and said, can you come up with some measure of the health of this particular attribute? In my case, it was benthic invertebrates. And like I said, a lot of the benthic bioindicators don't work for the upper estuary, but we came up with some indicators like diversity, native, non-native balance, and came up with sort of a historical trend to say how we're doing. The answer is complicated. The answer is always complicated, but it's okay. As I said, the problem is we don't have a lot of unimpacted sites to compare ourselves to. But for example, this is one of the sort of sub-indicators. And like I said, in Sassoon Bay, the number of native individuals dropped sharply off a cliff in the mid-'80s. But in the confluence of the rivers in the Delta, it's been okay. So you could read that since more non-natives tend to be a sign of a more impacted ecosystem, this is sort of a way of looking at the benthos and saying, how are we doing? We're doing okay. Mixed signals, depends where you are. And then this is sort of the latest, and to my mind, one of the more interesting efforts that the benthic monitoring has been going to. This is a Drought MAST team, MAST standing for Monitoring Analysis Synthesis Team. And this is a multi-agency group that has gotten together to basically say, what does the drought look like out in the Delta? We all know that drought is less water. Well, how does that manifest? And so what we've been doing is getting together a lot of different abiotic and biotic data, including some of the clam grazing numbers, and saying, how does drought stack up differently? You know, if you're in the Delta, living in the Delta, how does it look different as opposed to non-drought years? This is in process right now. We're hoping to get a manuscript out sometime by the middle of the year. And there will be, I think there's going to be a talk on this also at the IEP conference, so keep your eyes open for that. But this to me is a very exciting effort. And again, little sneak preview. Drought looks different. Yeah, the really dry years do look different than the really wet years. So check in for more details as this comes out later. And that's pretty much all I wanted to start with today. Hope you guys know a little bit more about the benthos, and if anyone's got questions, I'd love to take them. Yeah.
For your long-term data set,
yeah.
For your long-term data set, what would be the depth range that these data represent? All the way up into the marsh, all the way down to the bottom of ship channels, or somewhere in between?
The minimum that we can go into is pretty much what a whaler can get into. So I would say our minimum is somewhere around 5 or 6 feet, because it's harder to, we can't reliably get to those sites if they're much shallower than that. And the deepest, I mean, we go to Carquinez Strait, so we're going down 70 feet maybe.
Up in the Delta proper? Sorry, in the Delta proper?
Up in the Delta proper, we tend to, the furthest up in the Sacramento that we go is up to about Rio Vista. We've gone further up in the past and for special studies. But again, it becomes a question of are we getting more data as we go further upstream, or are we getting new data? And the answer is kind of no, because it seems like if you go much upstream of there, you really have to go to more stream monitoring tools, and that's just not what we have done. I know other people have done sort of stream monitoring, but they're different methods. You tend to do dip nets or hand cores and stuff like that. We don't go into the vegetation. I wish we did. If I had a big pot of money, I would love to do some in-vegetation monitoring sort of among the submerged aquatic or the floating aquatic vegetation as a complement to our benthic stuff, Deponar doesn't work, especially in heavy vegetation. It's sort of a little bit here and there we can manage, but in heavy vegetation, we just can't get down there. You need different methods. I would love to know what happens as you get closer to the shore and shallower and in vegetation, but right now, we don't have that. All right. Thank you all so much.